Good afternoon. Speaker number 50 and 1 hour after lunch. I think I have a challenge. But I'll definitely stay within the 10 minutes. So, public company, Brussels stock market. We are a company that is focused on diabetic eye disease. More specifically, we try to develop VEGF-independent molecules to treat DME. Currently, we have 3 molecules in the clinic, 2 of them VEGF-independent, and then 1 VEGF pathway. I think we have a proven ability to develop a product, not so much to sell a product, Jetrea. And with currently over 60 million cash at the bank, we're okay for the coming 2 years. So what we try to do basically is, like most of us try to do here in this room, improve on what is excellent therapy, anti-VEGFs. But we do believe that a faster onset of action and a higher responder rate is definitely to a benefit. Secondly, we do believe that long-term suppression by anti-VEGF is not a good thing because it does block, next to pathological, also physiological blood vessel formation. And thirdly, the durability of treatment is, of course, very important. In general, improve the treatment outcome, decrease the treatment burden. The molecules we have, the 149, is basically an inhibitor of plasma kallikrein. With that, we try to do something for suboptimal responders to anti-VEGF. The integrin inhibitor 687, there we think we have a molecule with a broad therapeutic approach that also beyond DME can be showing activity. And then thirdly, the antiplacental growth factor. With that, we think we could try to complement anti-VEGF therapy. If we look at our pipeline, first of all, the PCAL inhibitor, we had top-line data mid of the year and plan to start the phase 2 next year. 687, the integrin inhibitor, we come out with top-line data by December, end of December, and phase 2 also next year. Then mid-year, we showed some 317 anti-PLGF data, some mixed data, to be honest, and that project is under review. We still have 1 trial running with anti-PLGF, that's MACTEL-1, and there early next year we should have the top-line data. The PCAL inhibitor is basically a collaboration with Bicycle Therapeutics from the UK. It's a highly potent and highly selective and stable molecule. Clearly, PCAL is described as a well-validated target for edema, inflammation, and the prevention of microhemorrhages. So the phase 1 study that we announced middle of the year is basically ascending dose, 525, 125 microgram, and standard design. So what we saw is, first of all, we saw an increase of the BCVA day 1 of more than 3 letters, going up to 6.5 day 14, and even after 1 injection, 90 days later, we got 6.4 letters. I think these are very encouraging results. It was a safety study primarily. There were no DLTs. There was no ocular serious adverse events. And the only side effect we had was related to the injection procedure itself. Interesting, the BCVA gain was immediate and sustained until day 90. Secondly, the 687, the integrin inhibitor, that is basically a molecule that we licensed from Galapagos. It's a molecule that we know works in the upstream and downstream of the anti-VEGFs. But what we found is that it not only influences multiple processes, but it can also have effect beyond DME, namely wet AMD. So far, in our hands, we could not make it work in the dry AMD space in the preclinical environment. This is our preclinical data on cynomolgus monkeys. And we clearly see that with regard to the angiogenesis inhibition, we play at least as well as Lucentis. Phase 1 is currently running. It's a US-based study, 4 ascending doses, enrollment complete, data as said, end of December. Finally, 317, antiplacental growth factor. That is basically part of the VEGF family. Interesting always has been that in severe cases of DME, the levels of PLGF are increased. What we saw in our phase 1 data at the 8 milligram, we saw up to 6.8 letters improvement. And there was a certain durability because basically month 5, so 3 months after last injection, we still had 4 letters improvement. After the phase 1, we initiated phase 2 in combination with Lucentis. And there what we found is that in overall, we didn't find a difference between monotherapy and then the combination therapy. But what we did find was some interesting trends with regard to some subpopulation that in the less than 65 letters EDTRS, we found there's a certain tendency for those patients to respond better, as well as in the group of the poor to non-responders, also there, the combination small trend in favor. Now, either way, what we decided basically is to still wait for the MACTEL data, which will read out early next year. It's a rare disease, unilateral blindness in male. Based on that, we may or may not proceed with that program. So in conclusion, we basically have these 3 molecules, but we think to move next year with 2 molecules. First of all, the plasma kallikrein inhibitor, novel VEGF-independent pathway, positive phase 1 data with immediate effect, but also sustained effect. Secondly, 687 pan-RGD integrin antagonist, VEGF-independent largely, readout by the end of the year, and then 317, we'll see. Start of the phase 2s, pending data, of course, on the integrin inhibitor is early next year. And with the 67 million, we have enough money on the bank to finance those. Thank you.